Okay, final part, mutations. So you can imagine that, well, let's just go. So mutations, as we've covered before, are changes in the genetic information of a cell. And so there can be maybe one nucleotide wrong, or maybe a few. But we're going to focus mostly on point mutations. So changes in just one nucleotide pair in a gene. So remember, there is a double-stranded helix. And so if there's one letter wrong, the other one is going to still be complementary. And so it's one pair of nucleotides that is wrong. So point mutations are changes in one nucleotide pair. So there's a mutation at one point. And so even the change of one single nucleotide in a DNA template can lead to the production of an abnormal protein. So if a mutation has an adverse effect on the phenotype of the organism, then the condition is referred to as a genetic disorder or a hereditary disease. So the classic example is sickle cell disease, and this comes from a point mutation, so just one nucleotide pair that is wrong. So on the left is the wild type beta hemoglobin, so just the normal hemoglobin. And so this is the nucleotide that we're the codon that we're focusing on. So it's supposed to be CTC on one strand and GAG on the other strand. And then so when the mRNA is transcribed, this codon is GAG, and that codon codes for the glutamine amino acid, glutamate. And so in this sickle cell mutation, it's flip-flopped. It's the wrong, it's T and A, but on the wrong sides. So now this is CAC on this strand, and this is CTG on this strand. And so when the mRNA is transcribed, this codon is totally different now. Instead of GAG, this codon is GUG, and that codes for a totally different amino acid, which is valine. And so that leads to a totally different shape in these red blood cells because of this hemoglobin being different. It doesn't fold properly. And so just one nucleotide difference will lead to a totally different shape of the red blood cells. So they're supposed to be kind of like nice and round and happy. And in this case, they look like a sickle. So kind of like to cut wheat or whatever. That's why it's called sickle cell, because it's shaped like a sickle. And so that came from just one mutation. And then that person might have a host of symptoms with that because of that. So point mutations within a gene can be divided into two general categories. There could be a single nucleotide pair substitution, or there could be nucleotide pair insertions or deletions. So this is an example of a substitution. So like the GAG, the A is substituted with a T. It's not like anything new was inserted or deleted, messing up this frame. It's just a substitution at that amino acid, or at that nucleotide pair. So that would be a single nucleotide pair substitution. But I'll talk about examples of the other ones. So a nucleotide pair substitution replaces one nucleotide and its partner with another pair of nucleotides. And that can lead to different effects. So a nucleotide pair substitution can happen, but then it might lead to either a different amino acid, it might happen to lead to the same amino acid, or it might just disrupt everything and stop translation altogether. So there are additional terms that we can add to these mutations based on the effects that they have. So silent mutations have no effect on the amino acid produced by that codon because of that redundancy in the genetic code and how there can be multiple codons with different nucleotides at the third position that end up coding for the same amino acid. So if there's a nucleotide pair substitution that has no effect on the amino acid that's made, then this is called a silent mutation. As compared to these other versions, so say that there is a substitution, and it still codes for an amino acid, but it's not the correct amino acid, and then in this case, it's called a missense mutation. It kind of makes sense, like it still codes for an amino acid, it's just the wrong amino acid, so it makes missense. And then the last example is nonsense mutations. So in this case, there's a change in the codon, where instead of coding for an amino acid, it just codes for a stop codon. And so this leads to like nonsense, like you disrupted this whole protein because it doesn't even make an amino acid, it's just an early stop. And most of the times this leads to a non-functional protein. So I'll show some pictures on the next few slides. So this is an example of a silent mutation. So on the top are the normal wild type DNA, and then mRNA, and then series of amino acids. So we're focusing on this area right here. So if the DNA here is wrong, or well, is correct, sorry, so this is the wild type, right? So on one strand is CGA. My God, I'm losing it. Okay, CCG is what we're focusing on. And the other strand is going to be GGC. So the template strand, CCG, we're going to transcribe GGC. And then that codon codes for glycine. And so if there's a mutation here, and it happens to be in the third spot of a codon, it might not make a difference. So even though this is wrong, it's supposed to be GGC on this strand and CCG on this strand. If there's a mutation and now it's incorrectly CCA on the template strand, that means the mRNA is going to read GGU instead of GGC. But luckily in this case, that codes for the same amino acid. So even though there's a mutation, you almost can't tell because it codes for the same amino acid. So then it's called a silent mutation. A missense mutation is when it happens somewhere else. So if the mutation was earlier in the first or second nucleotide in the codon, it would more likely lead to a different amino acid outcome. So if the mutation happens in the first or second position, it changes the amino acid. So again, in this case, the mRNA is supposed to read GGC. In this case, because there was a mutation in the first letter, this is a totally different codon, AGC, and that codes for a totally different amino acid, serine, instead of glycine. So this is a missense mutation, like it makes missense because it's the wrong amino acid, but at least it's still kind of making sense, like you would proceed with the same rest of the protein. Say this was a lot longer, you know, it still would mostly be the same outcome. But then here, say there's a mutation somewhere and it leads to an early stop codon. So now we're looking at a different area. So if here it's supposed to read AAG, the mRNA now reads UAA because of this mutation here. And that happens to be an early stop codon. So that just makes nonsense. This protein is nonsense because it's all messed up. It started and then stopped. So this is kind of like an extreme example, but yeah, if like a protein is supposed to be very long and it has a really early stop, this is just not going to be functional anymore. So that's why it's a nonsense mutation, as opposed to a missense. Like, it's wrong, but hey, it didn't totally mess everything up. So those are different kinds of single nucleotide pair substitutions and the effects that they would have. And then the other kind of mutation that can happen are insertions and deletions. So these are additions or losses of nucleotide pairs in a gene, respectively. Insertions, there's an additional nucleotide pair, and then deletions are the loss of a nucleotide pair. And so these can have disastrous effects on the resulting protein more often than substitutions do, because this messes up everything. So insertion or deletion of nucleotides can alter the reading frame. So if, normally it's supposed to be read this codon, this codon, this codon. If you insert or delete a nucleotide, that shifts everything. That shifts the codons to be totally wrong. And so I'll show a picture. So this is what is called a frame shift mutation if it messes up the reading frame of all of the codons. And then insertions or deletions outside of the coding part of a gene could affect how the gene is expressed. Oh, I guess there's no... I thought I had a figure. But say, for example, this is the wild type. Say all of a sudden there was a T inserted between here. Now, this template strand would be TTA if there was a T inserted. Then that shifts everything. So this isn't even a methionine anymore. That's not a start codon. That would be AAU, and that's totally different. And so that would mess everything up. So that is what insertions and deletions would do. It would change the whole reading frame, making a frame shift mutation. And so remember from last lecture that spontaneous mutations can happen during errors in DNA replication or errors in recombination or due to physical or chemical agents. And so those are called mutagens because they cause mutations. And there can be chemical mutagens or physical mutagens. And so most carcinogens or cancer-causing chemicals are mutagens. So like the mutations that they cause will happen to cause cancer. And most mutagens are carcinogenic. And so as I mentioned, when I did the gene editing, the actual technique I used was called CRISPR-Cas9. And so CRISPR-Cas9 is a really cool tool. So biologists who study disease-causing mutations have looked for techniques for gene editing to alter genes in a specific way. So when I did it for my purposes, it wasn't for studying disease-causing mutations, but this is a tool that other people can use for different purposes. And so the powerful technique called CRISPR-Cas9 is transforming the field of genetic engineering because we can do gene editing in a much better way, basically. So normally in bacteria, the protein Cas9 acts together with a guide RNA to help defend bacteria from viruses. So remember, bacteria can have their own viruses infect them. And so normally in bacteria, this mechanism is to help defend bacteria from getting virus infections and having their viruses inject DNA. And so, yeah, there's a protein called Cas9 that cuts nucleic acids. And then there's a guide RNA that helps that protein recognize things. So the Cas9 protein will cut any sequence to which it is targeted. So let me do a drawing. Let's make it blue. So here's a blob just to represent the Cas9 protein. And so this protein enzyme will cut sequences. It could be DNA, it could be RNA. It depends. There's actually different Cases. So Cas9 protein will cut whatever that RNA, the guide RNA it has, tells it to. So based on what bases the guide RNA has on it, it's going to look for complementary stuff. And whatever it finds and binds to, then the Cas9 protein will cut at that site. So it's like looking for that sequence, say A, C, G, whatever. It's gonna like scan around the DNA, look for that spot, and when it finds it, that's where Cas9 is gonna cut. So scientists can introduce the Cas9 and guide RNA complex into a cell that they want to alter. That guide RNA is engineered, so you tell it what to look for. It's like a little homing device. You tell it what gene to look for, and then it'll look for it, and Cas9 will cut both strands of that gene. And then once that's cut, you can insert or delete or do different things. So the broken ends of that DNA, once it's cut, trigger a DNA repair system. The cell wants to like, whoa, we don't need a broken end. Like, that's bad. We got to fix it. So the repair enzymes might remove some random nucleotides, or they might add some random nucleotides while fixing those broken ends. Like, they're trying to fix like a railroad track. They're just like, oh, maybe we should cut out the damaged parts, or maybe we should add in more, and then stick them back together. And so this is a way for researchers to either knock out and disable a given gene, and then you can study what it does. So maybe in that experiment at the very beginning, instead of just randomly putting x-rays on the mold, say you wanted to study a specific gene, you could use CRISPR-Cas9 to look for that specific gene, disable that specific gene, and then study what it does, instead of just randomly mutating and then seeing what happens, and then having to learn which gene that happened in, and so on. So it's super interesting. So to treat genetic diseases, researchers have modified this technique. So you're not knocking something out to learn what it does. That's kind of unethical, especially in humans. But to be able to treat genetic diseases, we can use this technique too. So they can introduce a template with a normal copy of this gene into somebody that has abnormal copy. And then hopefully that helps replace this abnormal copy with the normal functional copy, and then corrects this defective gene. So in this way, yeah, the CRISPR-Cas9 system can edit the defective gene and correct it in this person. So this is a figure showing basically what I drew. So here is the blob that is representing Cas9. And so here is the guide RNA that guides Cas9. It'll look throughout the DNA for the complementary sequence, and then when it finds it, Cas9 is going to cut. And when it's cut, the cell will try to fix it. So either it will kind of fix it with random nucleotides, or if you give it something like the normal functional copy of a gene, then it'll replace it and fix that. So say, for example, the sickle cell, the trait for sickle cell disease, it might be able to, make a break here and then fix this sequence back to the correct one. So that's CRISPR-Cas9. And so treatments for some human genetic conditions like sickle cell disease have shown promise when tested with mouse models. But there are still concerns about using this technique in humans because it's not always 100% perfect. There is the possibility of unintended effects on genes that have not been targeted. So even though you're trying to specify as much as possible, like, hey, this is the sequence you should be looking for, sometimes it messes up and it cuts at the wrong sequence. And so we don't want that happening in a human. So there's still a lot of concern about using it in humans. So biologists have agreed to use extreme caution as the field moves forward, and that's why I was able to use it in tiny worms that are microscopic and not humans to study like neurobiology and dopamine receptors. Yeah. So again, revisiting the question of what is a gene, what does it code for? It's evolved through the history of genetics. We have considered a gene as a discrete unit of inheritance, a region of specific nucleotide sequence, a DNA sequence that codes for a specific polypeptide chain. And so now to give a final version, a gene can be defined as a region of DNA that can be expressed to produce a final functional product that is either a polypeptide or an RNA molecule, because sometimes it could be an RNA molecule that you're trying to make, actually. So yeah, we've come to learn a lot, and the field continues to grow. There's still things that we don't know and are still figuring out. So that's it for this lecture. These are all the lecture goals and objectives, and hopefully these are the things that you can do now with all this information.